This video covers the fundamentals of recombinant DNA technology. In a way, you can think of this as Genetic Engineering 101. So after this video, should understand the basis of recombinant DNA technology, what it means and how one may clone a gene, how we can create genetically modified organisms, and I'll try to illustrate the applications of these techniques using the cystic fibrosis transmembrane regulator gene, a gene that we have talked about in previous videos. When people talk about recombinant DNA technology, they're usually talking about what we call molecular cloning of a gene. So the whole idea behind molecular cloning is that you take a gene of interest, which is basically a segment of DNA, and you're going to somehow isolate it from the rest of the DNA in a particular cell or organism. And then you're going to take this isolated DNA segment and clone it into a vector. So the key words here are vector, cloning, and in this case we will talk about a type of vector called a plasmid. So what this illustration from the Campbell textbook shows is the insertion of a particular DNA segment into a plasmid vector, which generates a recombinant DNA molecule. So a plasmid is a circular DNA molecule, in this case found in bacterial cells. It can be isolated from bacterial cells. It can be cut open or linearized and then ligated together or spliced together with a foreign DNA segment to generate a new or novel DNA molecule that contains a new combination of DNA sequences, and that's what we mean by recombinant DNA. And the whole purpose of creating this recombinant DNA construct in a vector is that this can then be propagated or maintained in a cultured cell. And by culturing these cells, it's then possible to extract or re-isolate this vector DNA containing the cloned segment of DNA for further manipulation. So the definition then of a vector in terms of molecular cloning or recombinant DNA technology is a molecule that can carry or propagate a targeted DNA segment of interest in living cells. And the most common vector used by molecular biologists and genetic engineers is the bacterial plasmid. So what's more difficult is actually obtaining that gene of interest. How to find that segment of DNA in that one segment of DNA in the entire genome of an organism that you're interested in, that encodes the protein that you're interested in studying, or that contains a promoter sequence. So since all DNA is chemically similar, they all consist of A, C's, T's, and G's, how do you find that one DNA segment, maybe 3,000 or 4,000 base pairs or so, from an entire genome that may be several millions of base pairs if you're talking about bacterial genomes, or vertebrate genomes having billions of base pairs? One way, it all gets... Okay, so that's actually a topic that's beyond what we can talk about in this class. But the problem of finding and obtaining this gene of interest is vastly simplified if the genome of the organism has already been sequenced. And given the DNA sequencing technologies of today, it's fast becoming realistic and feasible to actually sequence the entire genome of whatever new species you're interested in working with, and then you'll be able to find the gene of interest in the genome sequence. And once you know what sequences you're actually looking for in this already sequenced genome, then we have a number of options. One is to look for what we call restriction endonuclease cleavage sites in the DNA. And we will talk about that next. And these restriction endonuclease cleavage sites will allow us to precisely cut DNA at those sites. And if there are appropriate sites available, then we can use restriction enzymes to cut the DNA at those sites. And those DNA segments are actually ready to ligate into whatever vector, plasmid vector or other vector, we would like to clone it into. If we do this with genomic DNA, then what will we get are relatively large fragments containing introns if we're talking about eukaryotic genes. If we're talking about bacterial genes, then we don't have to worry about introns. Since in our example we're going to be talking about CFTR, and the size of the CFTR gene in the genome is about 189,000 base pairs. So that's very large indeed and contains numerous introns. And that actually makes the restriction endonuclease site or method impractical for cloning the CFTR gene, because that 189,000 base pairs is way too large to actually clone into a plasmid vector. Even if we could ligate it or insert it into a plasmid vector, there is no way that we can actually transform it into a bacterial cell or somehow stably maintain and propagate it in a bacterial cell, because there are limits to the sizes of plasmids. So while that's a possibility for much smaller segments of DNA, and maybe for bacterial genes, it is not a feasible method for cloning very large segments of DNA. Then a second method would be to make a cDNA, a complementary. So cDNA stands for complementary DNA. And it's called complementary DNA because it's complementary to the messenger RNA. So if we're still interested in cloning the CFTR gene, the messenger RNA for CFTR contains all of the coding information to make the CFTR protein, but it's much shorter. It's only a little over 6,000 nucleotides long, the messenger RNA. But we can't clone messenger RNA directly. Instead, we have to convert the messenger RNA to a DNA, a copy DNA sequence or complementary DNA sequence. So it will be the complementary DNA sequence will have no introns and will be much more compact at only a little over 6,000 nucleotides long. And we can actually add restriction endonuclease cleavage sites to the ends of our cDNA to facilitate cloning. A third method that we have is the polymerase chain reaction, or PCR. Now, given the size of the CFTR gene, it's impractical to PCR genomic DNA. So PCR is the polymerase chain reaction. And while I'm not going to take time right now to go into the details of PCR, it's a method of making many millions of copies of a targeted DNA sequence. The limitation of PCR is that there is a limit on size, practically less than 10,000 base pair DNA fragments are amenable to PCR amplification. That sort of rules out genomic DNA for CFTR. Now, if we wanted to clone a bacterial gene, then it would certainly be feasible to PCR amplify the bacterial gene from bacterial genomic DNA, but it's not feasible to PCR amplify a 189,000 base pair CFTR gene from human genomic DNA, because that exceeds the size limits of PCR. However, we could start with messenger RNA, and then we could use reverse transcriptase to first make a cDNA, and then do PCR amplification of the cDNA. And we can incorporate restriction endonuclease cleavage sites into our PCR primers. Okay, so let's, before we get too much further along, let's talk about restriction endonucleases. Restriction endonucleases, and here this figure shows EcoR1, which is one of many restriction endonucleases found and isolated and purified from many different species of bacteria. So there's hundreds of restriction endonucleases that are known and available to the molecular biology toolkit. What all restriction endonucleases do is they cut DNA at specific sites. Each one has its own specific site that it cuts. EcoR1, isolated from E. coli, scans the DNA for this particular sequence, GAATTC. You will note that it is what we call a palindrome, because GAATTC on the top strand, if you look at now the complementary strand also goes GAATTC, okay, 5' to 3'. So the sequence reads the same forwards and backwards, and that is the definition of a palindrome. So most restriction enzymes cut at particular palindromic sequences. EcoR1 cuts wherever it finds this GAATTC, and only where it finds this GAATTC. And when it finds that sequence, it makes a cut between the G and the A on both strands. And so the resulting cleavage product of EcoR1 are these molecules that contain these ends. So as a result of an EcoR1 cleavage of DNA, what we get are DNA fragments whose ends have this AATT 4-nucleotide single-stranded overhang. So this AATT on this DNA fragment is complementary to the AATT on the other DNA fragment, and so it's possible for them to form base pairs, okay, and sort of stick together. And hence these 4-nucleotide overhangs are called sticky ends. And you will note that it doesn't make any difference what the source of DNA is. So this could be a fragment of plant DNA that's been cut with restriction enzyme EcoR1. This could be a fragment of bacterial plasmid DNA that's also been cut with EcoR1. As long as you have DNA molecules, two different DNA molecules that have been cut with the same restriction enzyme, they're going to have ends generated by the restriction enzyme, and these ends are going to have complementary sticky ends, which will tend to then stick together. Now you can completely reverse the cleavage by restriction endonuclease by adding a DNA ligase and ATP. So what DNA ligase does is it will repair these nicked sites in the DNA to give you back a whole DNA molecule. So let's be clear about what these two enzymes allow us to do. Restriction endonucleases are smart molecular scissors that allow genetic engineers to cut DNA very precisely at particular sites or sequences in the DNA. So you can take a test tube of DNA and add it a dollop of purified EcoR1 or any other restriction enzyme, and it will cut wherever it finds its particular recognition site in the DNA molecule, generating all these sticky ends. Now, if you take away the restriction enzyme and you have a test tube full of DNA molecules with these sticky ends, and now you add DNA ligase plus ATP, these sticky ends will reanneal and reform unnicked or intact DNA molecules. So we now have a way of cutting DNA very precisely and rejoining them to make whole intact DNA molecule. So now we have all the tools we need for splicing different DNA molecules together. And that is the entire basis of recombinant DNA technology. Taking snippets of DNA from different sources, if they're cut with the same restriction enzyme, they have complementary sticky ends, they will tend to join together, and then you can permanently ligate them together with DNA ligase, which will produce a novel DNA molecule with fragments, DNA fragments, spliced together from different sources. So that's cutting and joining DNA molecules to generate recombinant DNA molecules. Here is the technique of making cDNA, complementary DNA, from messenger RNA templates. The key enzyme here is reverse transcriptase. Reverse transcriptase is also known as an RNA-dependent DNA polymerase. So reverse transcriptase is a DNA polymerase, only it reads messenger RNA. So it uses messenger RNA or any RNA molecule as a template and synthesizes DNA that's complementary to the RNA template. The enzyme reverse transcriptase was first discovered in retroviruses. The most famous of the retroviruses, to ordinary people anyway, is HIV. So the human immunodeficiency virus that causes AIDS is a retrovirus, meaning it has the infectious viral particle contains an RNA genome, and the viral particle also contains a couple of molecules of the reverse transcriptase enzyme, and when HIV infects a susceptible human cell, it uncoats the messenger RNA, its RNA genome is copied into DNA by the HIV reverse transcriptase. Well, there are other reverse transcriptases that infect mice and other animals, and the reverse transcriptase from these sources has been cloned and produced in E. coli. So we have purified reverse transcriptase enzyme that we can use in our reactions. So we start with messenger RNA template. Okay? So if you recall, if you start with a eukaryotic messenger RNA, so this is eukaryotic. Eukaryotic messenger RNAs, if you recall, have at the 3' ends a poly A tail. At the 5' end, you should remember they have a cap. And in the middle, all their introns have been spliced out. Now, because they have a poly A tail at the 3' end, we can start with a oligo DT primer. So the primer is just nothing but a string of T's, okay, a DNA made with just, this is a synthetic DNA molecule containing just T's. This primer will anneal to the poly A tail, and then when we add reverse transcriptase, the enzyme reverse transcriptase, it will extend from the oligo DT primer. It will read the messenger RNA template from 3' to 5', and it will synthesize our first complementary DNA strand from 5' to 3', just like any DNA polymerase, except using RNA as a template. Retroviral reverse transcriptases, once they have finished synthesizing the complementary DNA strand, they also have an RNase activity where any RNA found in an RNA-DNA duplex is degraded. So reverse transcriptase will then degrade the RNA that has been partnered with the complementary DNA molecule. So now we have single-stranded DNA molecules, and then we can add a DNA polymerase and another primer, and DNA polymerase will synthesize the second strand. So we have second strand synthesis. And at the end, we have a cDNA, a completed double-stranded cDNA molecule. So let's talk about vectors. So any DNA that has been spliced into a plasmid vector can be replicated in bacterial cells. So there are different types of vectors. Some vectors are just cloning vectors. They will simply propagate whatever piece of DNA that has been inserted into the plasmid in a bacterial host cell. Then there are other vectors that have more useful properties, and such as expression. So what these vectors will do is they will allow the host cell, the host bacterial cell, to express the cloned DNA, meaning transcribe and translate the foreign DNA segment into a protein in the bacterial cell. Now, there are other vectors that are designed to maintain, propagate, and express foreign DNA segments in other hosts. So, a plasmid vector that's good for expressing or maintaining a cloned DNA segment in E. coli isn't necessarily going to be suitable for maintaining or expressing a DNA segment in mammalian cells or yeast cells or plant cells. So depending on the targeted host organism, there are specialized vectors that are designed and appropriate for the host organism and contain features to allow maintenance, propagation, and expression of foreign genes in the targeted host organism. So here is a little more detailed map of a plasmid for cloning and expression of foreign DNA segments in E. coli. Now, any plasmid vector has to replicate inside the host cell. Okay? So the replication function is provided by the origin sequence. So the ORI sequence is kind of like an origin for DNA replication in E. coli. And so this is an E. coli origin of replication. And this is actually an origin of replication that initiates replication of the DNA molecule more than once per cell cycle. So this plasmid called PUC18, because of this particular origin of DNA replication, is maintained in E. coli at dozens of copies per cell because it's replicated multiple times per cell cycle. So that's the bare minimum for a plasmid vector, is that it has to have an origin of replication in order to be maintained inside the host cell. Another bare minimum feature is it has to have a restriction site for cloning foreign DNA segments into the plasmid. Okay? And this is a restriction site that cuts the plasmid only once, okay? Meaning it opens up the plasmid, converts from a circular to a linear DNA molecule. Here, this little red segment called a polylinker. A polylinker is a set of 8 to 10, multiple restriction endonuclease cleavage sites, all placed right next to each other. And all of these restriction endonuclease cleavage sites do not occur anywhere else in the plasmid, they occur only here. This provides the genetic engineer a choice of restriction enzyme sites with which to clone a foreign DNA segment. Okay? So the cloning into the plasmid is going to be done at this site because this is where multiple unique restriction enzyme sites occur. Then if you want to express the foreign gene, then there has to be appropriate sites for transcription and translation. What you see in this PUC18 plasmid is that the cloning site occurs in the middle of a segment of DNA borrowed from the E. coli lac operon. Ooh. So here's the LAC-I gene, and if you remember, that encodes the LAC repressor. Here's the LAC promoter. Okay. This contains all the control sequences for initiation of transcription. Okay. So it has the LAC operator, promoter, the cap binding site to make transcription inducible in response to the presence of lactose or other inducer of the lac operon, and as long as glucose is absent. So the lac promoter provides a site for both transcription, and because the cloning site is actually right after the start of the beta-galactosidase gene, this is the same as LAC-Z, it provides a site to initiate translation. So any DNA sequence that's been inserted into the polylinker can be transcribed and at least begin to be translated. Okay. If the DNA segment contains a coding region which is in frame with the beta-galactosidase coding region, it's in the same reading frame after it's been cloned into this polylinker site, then you will get a protein that consists of just the very beginning of the E. coli beta-galactosidase enzyme, but most of it will be encoded by whatever DNA segment has been inserted into the polylinker. Alright, so that takes care of replication of the plasmid in E. coli, cloning a foreign DNA sequence, expressing it, meaning transcribing and translating the foreign DNA sequence in E. coli. What do we mean by selection? Well, once we have ligated, so we can cut this with a restriction enzymes, cut a foreign DNA molecule with the same restriction enzyme. We mix the plasmid, the cut plasmid, and the cut foreign DNA molecule in a test tube. We add DNA ligase, and we get a recombinant plasmid that contains the foreign DNA molecule. Okay. So gene X, and that's been cloned into our polylinker site. Okay. So this is all in a test tube. Okay. So we add DNA ligase, and we get a recombinant DNA plasmid. Now we need to get it into E. coli cells. Well, the easiest way to get it into E. coli cells is to run a transformation reaction. We take a batch of E. coli cells, we add this DNA, shake them up, and plate them out, and hope that the E. coli cells suck up this DNA and it gets into the E. coli cells. Well, the problem with this transformation procedure is that it's highly inefficient. A very, very, very low percentage of cells actually take up plasmid DNA. So what we need is to somehow be able to select only those E. coli cells that have taken up this plasmid DNA and get rid of the 99.999% that have not taken up plasmid DNA. So here what we see is that in this cloning vector, the BLA gene stands for beta-lactamase, and it confers resistance to the antibiotic ampicillin. So it confers ampicillin resistance. So E. coli cells that have taken up this plasmid will be expressing the ampicillin resistance gene and will be resistant to ampicillin. So if we, after our transformation reaction, we plate the E. coli cells in medium containing ampicillin, all the cells that did not take up the plasmid will die because they don't have this plasmid, and all the cells that grow because they're ampicillin resistant will be because all the cells that grow will contain this plasmid, making them ampicillin resistant. So that's one level of selection. There's a second level of selection that this plasmid provides, which is that, if you think about it, inserting a gene X into this polylinker, into this site, near the beginning of the beta-galactosidase gene, is going to disrupt the beta-galactosidase gene. So plasmids that contain gene X are going to be LAC-Z-. They're not going to have a functional beta-galactosidase gene. Plasmids that don't contain gene X, that left out gene X and just religated, these are what we call empty plasmids. So the original plasmid was restored without any insert. Well, those would make host cells ampicillin resistant too. So when we get our ampicillin resistant cells, some of them will contain the recombinant plasmid containing our gene X. Some of them will contain just empty plasmid that religated upon itself. How do you distinguish between them? Well, by testing for beta-galactosidase activity. Those cells that contain a functional beta-galactosidase have empty plasmids, and we don't want them. Those cells that do not have a functional beta-galactosidase are most likely to contain a gene X that disrupts the beta-galactosidase gene. Okay? And those are the cells that we want. So there is actually a test using a dye where cells that contain functional beta-galactosidase turn this dye blue, and you get blue colonies. And cells that contain a disrupted or non-functional beta-galactosidase gene are unable to act on the dye and remain white. So this is called blue-white screening. And so you pick the white colonies, and those are most likely to then have a plasmid. They're ampicillin resistant, so they contain the plasmid. They form white colonies, so they have a disrupted beta-galactosidase gene, meaning that they have an insertion in their beta-galactosidase gene. So this is merely one example of the kinds of clever tricks that genetic engineers have used to clone genes. So as I mentioned, transformation is one way to get a synthetic DNA construct inside cells. And that works okay for E. coli and other bacteria. But if we're thinking in terms of plant cells or animal cells, or worms or fruit flies, you can't just kind of throw DNA onto a fruit fly larva and expect it to somehow become transformed. You have to use some other techniques. And this is a nice diagram that shows some alternative procedures. One is to coat the DNA molecules onto microfine tungsten or even gold nanobeads, and then shoot them from a projectile gun. It's kind of like a micro or nano BB gun. And you just blast them through the cell wall or whatever protective outer covering that the cell may have, and somehow get them into cells that way. Another is to use a microfine needle and directly inject the DNA into the nucleus of a eukaryotic cell, for instance. Another is to use a nature's own delivery mechanism. Viruses are highly specialized and highly efficient at getting their DNA into cells. So if you can use a viral DNA vector and clone your gene into a viral DNA vector, then the virus can get your DNA into the host cell for you. And there are at least a half dozen other techniques, but I think this gives you some idea of the variety. In any case, once you're able to get the foreign DNA molecule inside a cell, what will happen at some frequency is that the foreign DNA molecule will then insert itself into the host cell genome. So nature appears to abhor ends of DNA molecules. If there's an end of a DNA molecule inside a cell, it will either rapidly get degraded, or it will insert itself into a host cell chromosome.